immunology, and cancer and other diseases. And so Jim, I first knew when he was at Berkeley. He then moved to Sloan Kettering, Memorial Sloan Kettering Institute in New York, and then back to his native Texas at MD Anderson, where he's leading this program, along with Pam Sharma, who is trained in immunology. She's an MD, PhD, trained in immunology, and then did medical oncology, and has been very involved with the clinical trials on the technology and the ideas that Jim Allison started. Leslie, as you know, has had a long 30-year career with 60 Minutes. She's one of America's best journalists, I think, and has won numerous awards, including 13 Emmys. I have to put my glasses on for this. 13 Emmys, a Lifetime Achievement Award, the Edward R. Murrow Award, which is very prestigious.
I'm delighted to be here. One of the great, I hope, gifts to all of you is I don't know anything about science. So I'm going to be starting as if I'm in first grade. I hope you appreciate that. First of all, I'm just thrilled to be interviewing both of you. I'm just going to jump in because I heard a word from Dr. Stillman, my ears up. He said, cured. He used the C word. And most doctors don't like to say cured when it comes to cancer. So cured? Explain cured, Dr. Allison.
Well, people have been hesitant to use the word cured because, you know, there have been incidental successes in people that with late-stage melanoma, where their tumors disappeared, but
and never came back.
It was 7 months, and fewer than 3 people were alive at 5 years. So, and since then, the good news is we can do better. Since then, another drug was developed against a different target, and the two work very well together because they're kind of different. There's a study that's about 9 years out now showing in the same disease, late-stage melanoma.
Dr. Sharma.
Yeah. So as an oncologist who treats patients in clinic, I think it's true that when you see a patient with stage 4 cancer where the cancer has gone throughout their body, right? It's not just in one place, it's in multiple places. We're reluctant a bit to say the word cure when we give treatment because we know advantage of your own immune system to treat the cancer. And the other thing is that your immune system can have memory responses in some of these situations that you'll hear about.
Is immune therapy like a vaccine? Is it similar to a vaccine?
Both of them involve activating the immune system. Vaccines typically are considered to prevent something coming on. So what we're doing is unleashing these other kinds of cells called T cells that actually go out and directly search through your body. T cells are very cool. I first learned about them as an undergraduate when they'd just been discovered and nothing was known about them. And I asked them also try to use T cells. What we do is unleash T cells in the body. What I've done is study step by step the molecules that are involved in regulating them. And I found this molecule's the brake and reason that it might stop an immune response to protect you from getting harmed by it before it eliminated a cancer. So I found a way to, not necessarily, cancer cells have antigens on their surface that can be detected, but normally it's a complicated process, but there are a number of other signals that an immune cell has to get activated, change in structures.
Not the immune breakdown.
Yeah, and so
If you think about it, your own body has mechanisms that stop your immune response. So if you get the flu virus, you'll have an immune response to the flu virus, and then the immune response stops, and you get rid of the virus for that period of time. Those mechanisms that stops the immune response, train the immune system, we can change that.
So take one step back. Is it hormones? Is it fluids? Is it
part of it. It's a series of cells that your body has developed to protect you, mostly against viruses, bacteria, things like that. Also cancer, if we help it a little bit, all held in balance by mechanisms that we're still trying to understand. They keep it, allow it to protect. The thing about it is
that's
that
probably important.
it can kill you, but it has all these protective mechanisms in so that it doesn't.
Well, if you have cancer up until now, you usually got chemotherapy. When you give your immunotherapy, what happens to people? First of all, is it a drip? Is it an infusion? And do people get sick?
It's an antibody, and just goes in. You sit for. I actually was interviewed by a journalist one time who had melanoma as she was getting the drug. There's
One shot?
Most of the time, it's 3 or more, but it can be 1, I think. But we won't.
Did
we
they
won't
go
there.
home and just lie there moaning and groaning? 'Cause
No,
they're so-
usually. I mean, some people that his oncologist a week later said, I'm in Denver and I feel great. And turned out his tumors were completely gone when he
Oh, my
came
God.
back. But in most cases, they're very, they're not the overwhelming sickness that you get from chemotherapy. And with the chemotherapy, basically, it kills tumor cells a little bit lower doses than it kills normal cells. And it's given a lot because unless you kill every last tumor cell that is needed. So
Well, you see patients, Dr. Sharma. Tell us what you see when you give this treatment.
So the immunotherapy drugs, they're very different from chemotherapy drugs. When you do get a side effect, though, just remember what before, when we're telling the immune system to act against the cancer, that's what the immunotherapy drug is doing. Your colon, so you can develop diarrhea. And these things can be managed. We definitely know how to manage them now, and are very good at making sure we stop the drug, and we give steroids as needed to control the inflammation.
Tell me if this is true, that when you first started trying these in the clinical setting,
what we knew from the mouse studies is we're initiating a response. You basically, the way you work at
wait, so
attacking tumors.
so you'd give your treatment and it wouldn't happen quickly?
Yeah, it could take a while to come along.
Okay.
In some patients, it might take a month or more in the early studies when it's given by itself. Whereas chemo, I mean, the chemo drugs are poisons.
I think it wasn't working in the...
Exactly, they were.
Well, tell her about that.
Well, they
kept growing?
Yeah, they would get bigger and bigger, and they said, "Oh, it's growing. We got to, they failed. We got to go to something else." So there were 2 things going on that fooled the doctors, and so that was called pseudoprogression. It was actually the immune system killing the tumor cells.
But the tumor kept
But there
growing.
was no tumor. But what was called a tumor was growing, but it really wasn't a tumor even. And so they'd used.
Patients stopped trying it because they thought it wasn't working?
Some did, I think, but people kept on, and there were studies that patients would go back to their home communities, and they'd come to New York, for example, from wherever, and be treated, and doctors said it didn't work, your tumors are still growing, they go home, get chemo, and then the word came back, it's working, and finally it was.
When they got home, they realized.
Yeah, and then they would go away, but
We're doing better. One key that we look at in the clinic is not so much just a CAT scan. We're not treating CAT scans, we're treating patients. So if the patients tell us they're feeling really great and they're doing better and they're starting to enjoy their life more.
So let's step back. You start your research, and despite that, you kept going. So talk about the way the general community that you live in thought you weren't going up the right tree and you didn't stop.
Well, I think what was going on is that as molecular biology, and so that made a lot of sense to do it that way.
To you?
To me too. I mean, I thought
Only you.
Oh, but to me, I mean, it would take care of it, and so we started working in mice as we worked in tissue culture, then worked out in mouse models what the different controlling things were. Sometimes I did many things, tickle mice and give them the same tumor and then compare it, and we'd get some things that would slow them a little bit. But when we first tried this CTLA-4, that's the name of the target, the antibody to that, the tumors just went away when we injected it. It took a while. Yeah, in the mice.
Wow.
And it happened with every tumor model. We were able to see all of those. And at that point I said, we may be onto something here,
But this idea of using the immune system has been around in the field of cancer research at least 50 years.
The first person to propose it was Paul Ehrlich, who was a biochemist in 1906, actually.
Took a while.
Yeah.
Yeah, but why? One of the very first stories I ever did for CBS News in 1972 was this, it was going to be the cure. they were going to do immunotherapy in 1972. Nothing happened. So what was different about the approaches that other researchers were doing at that point that changed?
I think that a lot of the other approaches were based on good ideas, but sufficient knowledge of mechanisms at the time. And so rather than just start throwing things at the cancer, what I decided to do was understand how the growth of a T cell was regulated, to find the molecules that did it, the pathways as best as we could tell. In other words, the buttons to push on.
We have to step back. I don't know. Does everybody know what a T cell is? I know you talked about it before, but I don't know what a T cell is.
It's a white blood cell that protects you. There are actually several kinds, but anyway, they protect you against, if you don't have them, as in HIV/AIDS, you're in bad trouble. Cancers, you get infectious diseases, things like that. They're the things that prevent that, and if you lose them, you're in bad shape.
And people have lost them.
Yeah, in AIDS, that's what it is. You lose those, you don't have them, because the HIV virus kills a particular kind of T cell, and then.
That's what you did. You studied the T cells in the beginning and learned the mechanism.
Yeah.
And there are switches that you figured.
Well, there's one. It's pretty easy to explain. There's one called the antigen receptor, and that's the things is to know what it ought to react with. It's kind of like the ignition car. Like I said, you've got maybe 10, maybe 100 million, we're not sure, different ones in your body. Each one, or small numbers, clones have a different switch, if you will. And so that we worked that out in the early 80s, that molecule, and then found out that it wasn't quite so simple. There was another one. It's kind of like the gas pedal, a molecule called CD28. And we worked that out. Anyway, the next one we quickly found was CD4, and it's the brakes.
Oh.
And we studied earlier trying to do each one, but they didn't work so well. But when we found the brake, we found if we took the brakes off, when there's a tumor around, the immune system would see that as foreign and kill it. All we had to do was take the brakes off.
Oh my goodness.
And
Finally.
you have T cells that can recognize virtually anything, but they're not because they're held in check. Or they get started and they stop because if it's a cell thing. But anyway, that was basically the idea.
Was there a moment in your lab, aha, the
Yeah.
great bathtub story?
Well,
Except you were in the lab?
There were some aha moments when we finally figured out what those molecules did, but there was a big, I looked at the mice that we'd inject with the antibody, and it was actually an experiment where we had a lot of mice, and half of it had been treated with it. They all had tumors, half of them the antibody and half something else. And I went in to actually look at the mice, and I saw the tumors shrink and go away in half the mice. I didn't know which ones were treated, but it was pretty clear to me that half of them, that was probably the experimental group and not the other one. And so that was an aha moment.
You're in the lab, you're working with mice, you eventually get to people. You had to get FDA approval first.
Well, that to people, though, was interesting because we did the experiment I'm telling you about in 1994. We published it in 1995. It wasn't until 2011 that it was approved.
Whoa.
By the Food and Drug Administration. It wasn't until about 1998 or 99 when we could finally find a company that was willing to help us by helping us make the drug that we needed to repeat in humans what we've been doing in mice. We couldn't use the same drug in humans. So we had to make a new drug.
Why couldn't you use the same drug?
Well, it was an antibody that we'd made, and it's a mouse protein.
Oh.
If you inject a mouse protein in a human, it's foreign, and it'll cause problems.
But it could ignite an immune response.
It could have. It would. It would. If we injected mouse protein in humans, it would to itself, and then you might get kidney disease because your protein
Might kill you, but you'd have an immune response.
So what we found was actually that it had a mouse that replaced the mouse genes with human ones. So those mice actually made human antibodies, and we could use those then to make the drug. And so that's what we did. The first trials were about 2001 or so.
Does the medicine still come from mouse altered?
A lot of them still do. Some of them are made strictly by genetic material, in tissue culture, but a lot of them still come from making them in mice. They're not necessarily produced in mice anymore. They can be made in cells in tissue culture. made like you make beer or something, you know. Actually.
I don't think maybe we're learning too much. Is this true? This is reporting. I don't know. I need to get my source to tell me it.
Well, when I was little, my mother was sick a lot, and you know, I didn't really know what she had, and I was called in once, and they said, "Your mother's very sick," and I was actually holding her hand when she passed away, and it wasn't until several years later, you know, I just knew she'd been sick a lot. I knew she had some burns on her neck and scars found out that she'd had lymphoma. And that was and then shortly after that, one of her brothers got lung cancer, and shortly after that, another one of her brothers got melanoma.
Oh, boy.
And so, and then and years later, when my brother got prostate cancer, and so, you know, you know, that's the reason. So I always thought, you know, grew up thinking, if I could, I'd like to do something about this, because I'd seen chemo. I'd seen cancer, and I'd seen chemo, and I'd seen radiation and surgery, and thought, we ought to be able to do better than this.
You know, we've all thought that, haven't we? We've all thought they have to figure out something better than chemotherapy, and at last. So what can you tick off the kinds of cancers that this therapy, that we know already this therapy works on?
Oh, God, I'll give some, and you give some. So melanoma, late stage, early stage, lung cancer, anal cancer, it works in, what?
Wow.
It's easier to name the ones that it doesn't work in at all. And
All right, let me ask it that way.
and there's two of them, and that's glioblastoma and pancreatic cancer.
Oh, pancreatic, come on.
And those are the ones that we've got to do something about, and we're slowly through the trial.
So, but do you think eventually that this will work in pancreatic and all cancers?
No, well, I think it'll work on some patients with all cancers. I think we'll be able to cure cancers. The numbers are getting up there. Things I mentioned earlier in melanoma now we're up to perhaps 80%. In lung cancer, there was a recent trial, an earlier trial of a type called neoadjuvant. Pam actually did the first trial of that sort with these drugs earlier on in 2006, but that's what our institute is all about. We know the rules. We know some of them it works better than others. We can learn from that, and the ones we're learning from the ones where it doesn't work, why it doesn't. And you know, the good news is we know we can do it in some cancers, so it's a matter of just working. In some cancers it will. But in other cancers, I think it can really be helped if you do a little bit of radiation and a little bit of chemo, but you don't have to try to kill every last tumor cell. You can just give one treatment, maybe kill some tumor and stop. And if you give the drug, you know,
Yeah,
in some
part
patients.
of the arsenal, as they say.
Yeah.
What's so difficult about pancreatic cancer?
Well, it doesn't have very many mutations. That's what's known as mutations. You know, you've got in melanoma, you've got a lot of mutations because it's caused by being in the sun and ultraviolet damage, lung, bladder,
That's
smoke.
easier?
They are easier because they got a lot of targets. I mean, it's kind of a surprising thing to me that the cancers which were the trickiest to treat by more conventional means because they had so many mutations that by virtue of those mutations could escape what you threw at it.
All right, I'm going to ask you if you want to write questions down. We'll be collecting them shortly, but breast cancer? Did you mention breast cancer?
Yeah, there is an approval in breast cancer as well. And again, it's combination with chemotherapy. So as Jim was saying, sometimes you don't have to give the immunotherapy alone, or you don't have to think that immunotherapy will replace chemotherapy. Sometimes it will be given in combination in certain tumor types. The other thing, you know, to remember is that you're pushing in the right direction to get that T cell going. But T cells are only one component of the immune system. And so the immune system has myeloid cells, natural killer cells, B cells. All these other cells are part of your white blood cell system, which are part of your immune system. And we still have to figure out the mechanisms of how each of these are working. And
In pancreatic cancer, what you asked about specifically, there are a lot of those myeloid cells that Pam mentioned, and those typically turn off T cells. They're made to function to protect wounds. And in doing that, they sort of keep the immune system from causing any further damage. So they'll actually turn T cells off. But if you treat them right, we know from some work that we've done in mice now that you can also do things, and we don't exactly know what's happening in the myeloid cells, but there's certain things that we could do. They'll help the T cells kill the tumor, so we can make friends of them,
Every kind of, yeah.
no matter what it takes.
So if you do have questions, I know there were cards given out, send them to the end of the rows, and somebody will pick them up, and then we'll get to them. You know, when someone wins the Nobel, they almost always have this great story of how they heard. And at least half of them, at least half the times I've heard of it, they hang up because they think it's a hoax. You know, come on. And so tell
They
us
don't answer, which is.
what happened when they called you.
Well, the stuff that I did was around, and so people every year for a while were saying, "Oh, you're going to get the Nobel." I said, "Stop it! I'm not going to get. No, I'm not worried." I certainly didn't wake up at 5 in the morning expecting a phone call ever. The
Cancer Research
the Cancer Research Institute, which is headquartered here, and does a lot to fund cancer research, especially training of young scientists. But she was getting their top award actually. And so
This is a sad story.
so we came in the night before. Son called and said, "Dad, you won the Nobel Prize."
Oh.
Yeah.
He got up. He had been getting up at 5 in the morning and watching. He knew and he saw it.
He suspected.
And so, well, the funny thing about it is, well, not funny, but nice thing about it. Well, first of all, the word spread very quickly then. And so within an hour or so, our hotel room was full of people and bottles of this and that inebriating substance, and including a Hudson bourbon, as I remember. Sort
Don't
of.
forget that.
But anyway,
I forgot that you
they had my office number. And I was here, not there, and so he called and gave them my cell phone number. So they called back.
Is your son here?
No, I think
No?
there he is.
Where are
There
you?
he is back there.
Sat with him last night.
Robert, I think he brought the Hudson bourbon actually. But anyway, he got to film the call.
Yeah, it's pretty exciting.
Yeah.
Well, I guess you were completely overshadowed with your award. How sad. You know,
It all went out the window.
I
I
haven't.
think she was going to be interviewed by Time magazine, right?
Yeah.
Because they are partners in working on this kind of immunotherapy, but they're also married, and I haven't told you that at the end.
Yeah.
So also I hear that when you found out, after all your work, that you got the FDA approval to go forward with clinical trials on humans, that you were at a very interesting place when you heard that.
Yeah, I was at a conference on melanoma molecules and molecules that attack the cause of cancer. Anyway, the precision medicine type, and I was there as sort of the token immunologist. I think there were two immunologists maybe in that there, and that had made the antibody. Would hear it. Finally, I got an email with these guys. They were in California. It was 11 in the morning, and they had glasses of whiskey that they were holding, and I thought, I guess that's it.
Were you there, Bruce? This is very exciting. This is a question, I'm going to ask a question that I hate being asked, but I'm going to ask it anyway, because if there's a good answer, you'll all be interested. Have any interests outside of this research that?
Well, a few. I play blues harmonica a little bit, so I play in 2 bands.
He's in 2 bands. I wanted you to hear that.
Well, they're bands largely of scientists and clinicians.
Oh, okay, forget that.
Not bad though. We still play the one's called the checkpoint.
Lee Nelson. I'm not kidding. So you want to tell him your instrument?
The harmonica. Very,
He
very
plays a
arcane.
harmonica. Some of us heard him play last night, and it's kind of jazzy. It's very exciting. A lot of scientists are good musicians, it's true. So I've been waiting my whole life, and I'm not a young chicken, and you all know that, for the cure. For the cure. I know you have too. We're right there, aren't we?
Yes.
People, some,
CRISPR, which is, gene editing.
Yeah.
Is it like a parallel track or?
it's a tool that could be used to improve T cells, for example. Some people grow instead so that the normal suppressor mechanisms aren't there, so they'll stay alive and keep killing tumor cells longer. That sort of thing, so it's very useful.
Is it an outgrowth of what you learned about the structure?
I think the targets are, yeah. Yeah, and it could also be used sort of thing.
Your basic fundamental research on how the T cells work, how to turn them on, how to turn them off, launched these other avenues for
Well, I wouldn't say exactly
attacking
it launched it,
cancer.
but it launched
Yeah.
a lot about the ways they might be used in cancer
Yeah,
specifically.
I think what
They-
Jim's work was definitely the way to think about cancer treatments. CRISPR is a technology that you can use for lots of different diseases. They're thinking of how to use.
cancer,
Yeah.
then they go right to the T cell
Yes.
right now. All cancers involve T cells?
I think so.
Yeah.
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 It
Okay.
used to be thought that, the idea was, cancer cells are so much like normal cells, they're not viruses, they're not bacteria. That's not enough. The immune system, there theoretically at least, but there's never just one mutation, there are always more than that. But, yeah, it's a pretty fine detection system that T cells have for knowing what's foreign.
Okay. Do we have questions from the audience?
System that T cells have for knowing what's foreign.
Okay. Do we have questions from the audience?
Oh, that's ongoing already. I don't know if some of you may have read the New York Times headlines about how personalized medicine, whereby patients were selected. These were patients.
And another way to do it is based on a lot of the stuff that goes on at Cold Spring Harbor. You do the genome sequencing, and then you can detect what's mutated in the tumor, and then there's certain computer programs that can tell you which were structures that were used for the COVID vaccine. You can just plug in those sequences potentially into that vaccine and therapeutically treat a person with the exact antigen. Any doctor in the U.S. can prescribe these things. They're
Think
approved
about
by the
that.
FDA.
I know everybody in this room either had cancer themselves or knows someone who did, who suffered with chemotherapy. And, the story I heard about your mother, and maybe I just heard it wrong, was that you saw her on chemo and that it was horrendous. And you said, that can't be.
I saw her brother on
Her brother on chemo.
But I saw him go from this, Marlboro-like guy, literally, to a shriveled guy in a tent with chemotherapy, going in his arm, and that was not pleasant either.
No. It's so exciting. It's so uplifting. And I'll tell you something, we need these good stories right now in our lives, all of us. Can there be other approaches beside the immune system that have been considered and might be considered again? I think that's the question.
Sure. I mean, the chemotherapy is curative for some kinds of cancer, and the targeted therapies where you inhibit the mutated. Who knows?
Who knows? But then it sounds to me like a combination
Yeah.
of different kinds of remedies, whatever.
Well, that's one of the things that we're trying to do is combine chemotherapy, whatever it takes. I mean,
Yeah,
it's the bottom
whatever
line.
it takes, and to make it easier. Is the same immunotherapy used for all cancers? And we discussed a little bit about
Yeah.
that. Eventually, the silver bullet.
So I think the immunotherapy space has definitely become broad.
Approval.
We do have
Until-
approvals in some settings, but not all the tumor types that we have the approvals for in the metastatic setting.
Okay.
It's always done the metastatic setting first, and then it works earlier because, experimentally, they figure if you've got terminal cancer,
Yeah.
you can take more chances.
Right.
But-
But now, we were asking, somebody asked, if anybody gets cancer, can they get your treatment? Not if it's not metastatic, and most
Yeah, the first patient I ever met was a 22-year-old woman named, that was in 2006, and now she has two they're still teenagers, I guess. But,
No, I think they're a little older, but yeah.
maybe a little older now, but anyway.
What did
Last
you,
time I saw them, they were teenagers.
so you were beginning to say the name of the drug, and it's, do it again.
Ipilimumab.
I can't say it, it's written down.
It's hard, it's hard. They all end in, because it's a monoclonal antibody, MAB, made in mice, murine, so it's MUMAB. And all of them that are made from antibodies are called MUMABs, but then whatever goes in front of that is gibberish. Ipi was, I don't know where it came from, but since it was invented while I was at Berkeley, I tried to convince them to put an H in front of it and call it hippie. Limumab,
What about,
but
let's call it the Allison.
yeah.
Those
Wait,
are the
they're-
drugs
they're
you're
on
seeing in the
commercials?
Oh,
TV
yeah, there's
commercials.
commercials all the time, all the
I
time.
haven't seen these.
Yeah.
I have one final question, a final question about a particular, yes, no, you don't know, or?
A group that's doing some studies in glioblastoma. It's a team of 5 institutions working on this to try and understand glioblastoma better. And what we are and maybe come up with different ways to target for that tumor to get an immune response. So those studies are still ongoing, and we have a long way yet to go.
You know, they used to say that if it was head and neck, that you couldn't take chemo because there was some barrier, and that's not the problem.
No.
There's no head and neck.
Yeah.
Well, we've finally met our hero, everybody. So thank you all.